Welcome to Click for Biology. As part of our study of ecology, we're going to be looking at the concept of biodiversity. Within this particular part, we'll look at two concepts. One of the distribution of individuals within an ecosystem, but we'll also look at ways in which we can measure biodiversity as well, and of course define biodiversity. There's a blank PDF of this presentation linked in down in the description. You can download that and add your own notes as we go along. So the term biodiversity is made up of two components. If we look at the original term, bio referring to living things, and diversity here is relating to the number of different types of living thing. And so the simplest part of biodiversity is to list the actual species which is present within the ecosystem of our study. In a small ecosystem, this may be relatively straightforward, but if we have something as complex as a tropical rainforest, describing the entire species list may be, in fact, next to impossible. We could do a good job, but getting 100% of the species is very unlikely. This list of species is what we call species richness, and that is just one aspect of biodiversity. The other part of biodiversity is the relative abundance of each of these species. Now, abundance is the number of, and of course, the number of individuals in a species is the population. And we've already studied quadrating techniques for determining population sizes. And so our biodiversity contains not just the list of species, but also their relative abundance of each of those species. In other words, we would need to calculate the population of each of those species for our measure of biodiversity. Let's just look at a number of the details. Although we have defined biodiversity, we did not mention distribution. One of the problems of a study area and of quadrating techniques, so this is the study area, and then of course you would know that you would need to make a grid system in order to find the population. However, when we do that and we calculate the average number of individuals per grid and then calculate for the total sample area, there are some assumptions made, and that is about what we call the distribution. And generally, it's the case that quadrating assumes that there's an even distribution, that the individuals of the population are spread out equidistant from each other. However, if we take a look at this diagram here, our sample site is here. And although the images are quite small, we can see that in the quadrats that the species A is concentrated into this zone here. And whilst it's still present over here, it's less so. And so the distribution is uneven. If we have many of an individual in a particular grouping, we call that a cluster. So the species is clustered. So when we do a study of a population and we are attempting to calculate biodiversity, we can take a few moments to discuss the distribution of the species within our sample area. You'll recall that biodiversity is made up of the species richness, which is the list of species present in the study area. And you can see in this particular little image here that we have 3 different types of species, species A, B, and C in my table, and a brief description of what they look like underneath. In a proper study, you would need to identify them very carefully. The second part is that we need to then calculate the abundance or the population size. So for organisms that don't move, like plants, quadrating is the best way. For animal species, there are more advanced techniques involving animal traps. But we'll not discuss those in detail here. So we get the abundance. And then we can then combine those in a table. And so here we have our quadrats. Here we have the relative abundance of A, species A, in the different quadrats. And here we have species B and species C. And so then we can use the total abundance and the species list to write descriptions of what the biodiversity of this location actually is. You'll notice that species richness is down here, but this column is not additive. 3 species overall. So we can write descriptions of biodiversity. If you study ecology to higher levels, what you would find is that these numbers can be combined in a calculation to calculate what are called an index or indices, and those indices can be used as a numerical quantification of biodiversity, but we'll not go into those calculations right now. Biodiversity made up of species richness, made up of the relative abundance, and we can use those to describe the biodiversity of ecosystems. Here's 4 questions. Take 3 minutes, pause the video, write down your answers, check the answers with those which are in the description of the video down below. And once you've done that, wonder if you could take a moment to subscribe to the channel, give us a thumbs up. Here's the playlist, and here's a link to the next video in the course.